Okay, Professor Dr. Elena Aisha Azizan completed PhD from University of Cambridge in early 2020, trained psychology and joined the Department of Medicine Care when in charge of identifying clinical examination level 3. Study in primary aldosteronism in Malaysia population and also even locally. Additional technician in CFS University Award, and 18 million team of Education Federation of Enterprise Society and the Young Investigator Award for the Education Enterprise and Society Conference. Okay, so please welcome my Dr. Elena Aisha for her presentation. Uh,
Sorry.
yes, your voice. Yeah,
Can you guys hear me?
yeah. Can you?
Okay.
Yeah.
Can you see my screen?
Yes, yes.
Okay. So it was just my luck to go after very established groups and by very amazing leaders, Prof. Effendi and Prof. Zaliha, but I tried to make the best for my group, which is the molecular insights to primary aldosteronism. So
Dr. Elena, can you please speak louder because I can hear your voice.
okay. I think this is the loudest I can be. Maybe I just talk louder. Is that fine?
Yes, yes, yes. It's fine.
Okay. All right. So yeah, so myself as the project leader, and then the other faculties members that are involved with this group is Prof. Nadira, Prof. Shikin, as the clinical endocrinologist, and then we have Prof. Rohaiza and Associate Prof. Dr. Nani as the surgeons, and Prof. Rosman to give the radiologist input. And then for national collaborators, we have Dr. Prof. Associate Prof. Dr. Azian and Prof. Rahman from UMBI, and Dr. Azrai, endocrine specialist in Hospital Putrajaya, and Prof. Nur Azmi Kamaruddin, who was previously here, now in IJN. And we have many international collaborators, Dr. Mankit and Prof. Bernard from University of Manchester, Prof. Maurice Brown from Queen Mary University London, and Prof. Michael Stawisser from Australia, Associate Prof. Miroslav from Czech Republic. So quite a good international collaboration going on with this group. So unlike the other groups, I'm just going to go a little bit into the research topic as to why you might be interested in joining the group for molecular insights to primary aldosteronism. So a bit on the research background. So 1 in 3 adults in Malaysia suffer from hypertension, and hypertension, most of you know, is from high blood pressure. Is a silent killer. This means that those who don't have proper treatment of their high blood pressure has an increased risk for not only heart disease but stroke. So a lot of comorbidities you can get if this hypertension is not controlled. And blood pressure is regulated by the renin-angiotensin-aldosterone system. So the hint is in the word aldosterone. So primary hyperaldosteronism, which this group is interested in, can cause hypertension, but it is vastly underdiagnosed. So this is where we come in, or this group comes in. So we're interested in primary aldosteronism. So primary aldosteronism can be caused by aldosterone-producing adenomas or aldosterone-producing cell clusters. And why we're interested in the molecular insights is because these APAs and APCCs harbor somatic mutations in aldosterone driver genes. So these following genes that we had found: KCNJ5, ATP1A1, ATP2B3. So these are ATPase pumps, KECANET1D, and CTNNB1. So some potassium channels, calcium channels, and related with beta-catenin. But what we found is that these APAs that have different genotype actually have different not only cell morphology but also clinical phenotypes such as gender bias and tumor size. So large KCNG5 APAs have the F-like cell morphology and secrete 18 hydroxycortisol, whereas small CACNet1 APAs of different morphology, the zona glomerulosa, which is actually the physiological zone that secretes aldosterone. And this is more common in the very small aldosterone producing cell clusters at older patients. So we are interested in trying to identify why they have this different morphology when they have the different genotypes, and what we do know or is accepted, but we don't really know, is that the cells in the adrenal zones, it starts in the capsule from stem cells in the capsule, which leads into progenitor cells, and then leads to the differentiated cells, which are the different zones, the zona glomerulosa, the zona fasciculata, and zona reticularis. So we want to not make this a dogma anymore, but we want to make it for certain and understand why does certain cells acquire these certain different mutations. Why is that? So we are lucky because aldosterone producing adenomas. So from our surgeons, Associate Prof. Nani and Prof. Rahaiza, we get these aldosterone producing adenomas because they is actually can be cured of the hypertension. The patients that have an aldosterone producing adenomas, when they have this
Excuse me, Dr. Lina can hear you. I can't hear your voice. Okay,
now
now it's
yeah.
okay.
Okay. So whereas normally for bilateral adrenal hyperplasia, you do not have the treatment, you have mineral corticoid receptor antagonist. This is a rare case where the bilateral adrenal hyperplasia, one side or maybe both sides, are taken out. And why would you want to take it out? Why not just control it with hypertensive medication? So meaning symptomatic treatment. Well, because primary aldosteronism, if you see in this bar chart on the right hand of the two pairs, always have a higher odds ratio of stroke, non-fatal myocardial infarction, and atrial fibrillations, even when you match it with essential hypertension. So it has a more higher comorbidity than just those with hypertension. So this is where my group comes into play, which is, does the heterogeneity among the APAs have a genetic basis? We had previously found that with the KCNG5 at least, that there is a big range in the molecular profile of APAs, and there was actually a 800-fold difference in CYP17A1. Now, CYP17A1 is for a gene encoding for the steroid enzyme 17-alpha hydroxylase, which should only be in the zona fasciculata. Not in the zona glomerulosa, where it physiologically produces aldosterone. And the histology showed that there is 0% of the F-like cells in one versus 100% in another. So there's a vast spectrum of not only the histotype, but also the molecular profile. And when we panned it out with the diameter size and age at surgery, you can see that there is a sort like of a boxing ring. You have the red big circles here and the blue square circles here, where the squares are actually male and the circles are female, so they seem to segregate. And then when you look at the mutant versus wild type in the percentage of compact cells, there seems to be mutant segregates here, whereas the wild type seems to be split up into two groups. So in Malaysia, so that was back in UK where I did my PhD. So in UK, in Malaysia, I wanted to continue. So with the help of pathologists, Prof. Tan, Prof. Isa, and also the science officer Maya, managed to look at the histopathology and genotype of the APAs, and actually found the new CACNet1D mutation here, which was then functionally characterized by people in Yerkes Strikes group, and they found that this CACNet1 D mutation did shift the voltage dependence. So it was a functional mutation. And similar to what we had found or in KCNJ5, they do seem to have a specific histopathological phenotype. Although surprisingly, these larger KCNJ5 mutant APAs actually had a lower proliferation rate. So even though they're larger in size, they had a lower proliferation rate. So we thought, what can this help? This is all much basic science. How can this help clinically? Well, actually, it can help clinically because you can have distinct genotype, phenotype, which could facilitate diagnosis. And this is an example of how it can. So, for example, in this one, we found that CTNNB1 mutations stimulated the Wnt activation and cause adrenocortical cells to de-differentiate toward the common adrenogonadal precursor cell types, which means that now it can be activated by hormones that was highly present normally when you're pregnant, and therefore you get a more aggressive phenotype when these ladies are pregnant, and you can then the output of the clinical is that you have to take note during the first trimester if the patient have high blood pressure, which normally in a normal pregnancy would be in the second or third trimester. You should question whether this could be caused by this sort of adrenal C10NB1 mutant APAs. But at the time when we published in, even though it's a high impact journal, there was questions because these other groups had said there were no association with pregnancy in any other cases. Why are our group only finding? And they were not persuaded that this link was an activated mutation in beta-catenin, and whether the association with the hormone receptors actually underlies a prevalent cause of primary aldosteronism. And they were questioning why do these CTNN mutant APAs in other cohorts were not to found to have the high gonadal receptor, which is LHCGR, or had the clinical presentation of onset at pregnancy or menopause. So even if you publish in high impact journal, you do still get questions and criticism targeted at your journal. So we had this question. So actually, we did not find the whole thought, the whole answer when we had published in NEJM, but now we do have the answer why did they not find what we found. So actually, in our 3 patients that we had submitted, they are among these patients here. It is 10 patients here, and they, on top of having a CTNNB1 mutation, had a GNA11 and or GNAQ mutation, which then meant that the aldosterone producing adenomas can present in puberty, pregnancy, or menopause. So you can see here we've added one more patient, which actually helped us to identify this double mutation phenotype, which is actually this patient who was a 12-year-old boy. So all the others were female, pregnant, and presenting at menopause. This 12-year-old boy who hit puberty, had large testicles, and he had that phenotype, and he actually had these 2 mutations. So when we looked from the basic science, we can see that there is additive effects of GNA11 and GNAQ on the CTNNB1 mutation. So we weren't publishing rubbish, so to speak, but what we found was that you actually had an additive effect from having the GNA11, and this explains why others did not find in the cohort, and we actually had included other cohorts in this paper from France, from Germany, so we know now that it is the answer. So enough about the research background. I'd just like to talk a bit about the students in the group. So we have Fatin Atira, who's an ongoing PhD student. So she is interested in the aldosterone producing cell clusters, so the APCCs I was mentioning, and how it can actually increase with age. So it could be that aldosterone producing cell clusters is not only related to primary aldosteronism, but also present in those who don't have a clinical primary aldosteronism phenotype. Okay. And then we have PhD student Siti Khadijah, who's interested in somatic mutations in the aldosterone producing adenomas and to find a biomarker for this to be able to cure this disease. We have Dr. Loh Hua Heng. This is actually under Prof. Nolila, who is interested in looking at primary aldosteronism in diabetes, bone health, and sleep apnea. We have Afifa, who's actually awaiting convocation, who looked into genome-wide association studies. We have Shahira, who actually managed to win herself the um, uh, this was the Young Investigator Fellowship Award in the APCH. So I am very pro sending my students to go out. And she managed to publish in a high impact factor. We have Dr. Muhammad Arif also who graduated, who looked at Malaysian patients and how many of them had high aldosterone renin ratio. We also have some MMed students who had graduated looking at vitamin D, and MMed Sci students like Gani, who has also graduated looking at the aldosterone renin ratio in Peninsula. And I have a few international grants which had allowed Kachin, who's a part-time student, also science officer here in the endocrine lab, to managed to do flow cytometry techniques in UK in Queen Mary University London for 1 or 2 months. And similarly, Nur Fatin Shazrin, I had another international grant which allowed her to learn CRISPR technology at University of Manchester. So our research goal as a group is actually looking at what is the precursors for this APA and APCCs, finding the stem cell, the progenitor cell, the hypermutable cell, and to answer why only some genes, so those 5, 6 genes, and actually in the adrenal is the top 5 organs to have the most somatic mutations. Why is it the adrenal always have mutations, and to find a targeted treatment of aldosterone related hypertension, because we are using 10% of KKM to treat hypertension. We need to do targeted treatment to have efficient usage of resources to prevent this high silent killer, high burden silent killer. And we need to do something about our underdiagnosis, because we only have less than 1% of diagnosis. So we need faster and better screening and diagnosis of hypertension. And we believe this is through characterization of biomarkers for the precursor cells of APA and APCCs. So just to put this all into perspective, there is a distinct cell morphology and clinical phenotypes of the different mutant APAs, and this suggests that there are different cell of origin. So here's the adrenal cortex with the different zones, and the dogma, so not the truth, but the dogma of adrenal cortex development is that it's an unidentified stem cell in the capsule. It migrates and then it proliferates and it transforms into the different zones. But we want to show how this happens, how is it done. And we hypothesize why there is so many somatic mutations in the adrenal is because there is maladaptation to our modern diet. So our modern diet, we like nasi lemak, KFC, all chronic high salt intake. And this modulates aldosterone production in ZG. And when it goes haywire, that's when you acquire the somatic mutations and inhibit centripetal migration. So just to really stress, the adrenal gland is among the top 5 organs in humans. This was recently published in Science 2019, and it's significantly enriched with non-synonymous mutations. So mutations that cause an effect on the protein. So moving forward, we would like to identify and characterize these cells in the human adrenal cortex and find the hypermutable precursor cells for the different mutant APAs and elucidate why this somatic mutation occurs. So if you have any interest in aldosterone or interest in adrenals or even interest in hypertension or primary aldosteronism, do come and join and drop me a line. Okay, thank you very much. If anyone has any questions, thank you.